Okay, good morning, everyone. Could you do me a favor, please? In the chat window, if you can hear me, please type yes. Okay. Okay. Okay, I'm getting a lot of yeses, so I'm assuming that at the time the technology is working okay. So welcome back, everyone. I know it's very strange circumstances we find ourselves in. I never thought I'd be talking to a computer screen for the rest of the semester. The truth is, we don't know how long we're going to be in this mode. Things are still very fluid, so I think it's safe to say, at least for the next several weeks, we're going to be unfortunately taking classes online. So we'll do the best we can to support everybody and get everyone through it. So let me just make a few comments about the technology that we're using. Obviously, we're on this application called Zoom. And you should, probably at the bottom left side of your screen, you should have a little icon for your microphone. That's set to mute right now, but during the class, if you want to speak up, if you want to talk in some way, if you want to ask a question, it would be okay to unmute that and speak. Feel free to do that. Also, at the same time, there's a video icon. If your personal computer has a video camera and you want to display your smiling face to everyone, that would be wonderful. It's not necessary, but it makes it more interesting when we can kind of see each other. So, I'm used to looking at all of you and seeing your facial expressions and getting a feel for whether you're understanding or not. Right now, it's very hard for me to do that because I really can't see you. So I'm going to have to rely upon you to help me know when you need me to slow down, when you need me to repeat something, or when you need me to try to communicate in a different way to help you understand something. Okay, it looked like everybody was able to find the chat window. Also, I'm not sure because your screen's probably a little different from my screen. Do you have icons that say yes, no, go slower, go faster? Do you have these icons? They may be in the participant screen where you can potentially raise a hand or lower a hand if you need my attention. Please type yes if you see the raise hand and lower hand icons. Okay, I'm seeing yes from Nicole. All right, okay. So excellent. Okay, so during our time together, if you need to ask a question or need to get a hold of me, please raise your virtual hand and I'll try to see that. But if it's clear that I haven't seen it and you're able to, go ahead and unmute your microphone and please speak up. That way I can know that you need some help or need some attention. You need me to answer a question or something. That would be quite okay. Okay, so this is very strange, gang. So please bear with us as we all get used to this new way of having a course. At this time, let's take a look at the revised schedule that I sent out through email to be sure that everybody understands what that's all about. Okay, so at this time, I just want to go over this and make sure you know what's happening. So for the foreseeable future, we're going to be holding classes online just like we are now. If you know a classmate that should be in class but's not in class, I'd ask you to please reach out to him or her and make sure that they understand how class is going to be handled right now. Or at the very least, please ask them to send me an email so I can help get them online, because it looks right now that we have about 24 folks online with us, and that's great, but that's not the entire group. So if you have friends that are not with us, please reach out to them. Okay, so all the lectures are going to be recorded. I'm recording this right now. And at the end of each lecture, before the end of the day, I will send you a link to YouTube that has the recording of what we're doing right now. That way, if you want to go over something a second time, or if you missed a lecture because something else is happening in life and you just can't be here, you'll be able to go to the YouTube website and look at specific videos and review the content that we talked about. So those are going to be recorded and the links will be sent to you each day through email. Okay, also, you know, we can't have a normal lab experience as we're used to doing, but I am going to hold office hours about an hour for about an hour after the end of lecture. So starting at 9:30, I will continue online with you and to provide support for any questions you may have about lecture content, and also to go over the answers for your lab homework, okay? You are not required to be there if you don't want to be, but I would encourage you to maybe show up for at least one session on Monday or Wednesday so that you're aware of things that are happening in the course. For example, today I will go over the homework answers for work that was due today in lab, and I will also mention to you that I'll be emailing you today a histology packet, which will be some additional work that I'm assigning to help get you ready to identify tissues for the upcoming lab exam in about 2 weeks' time, okay? So there will be some new pieces of information for you as you stay online with us for the lab portion of the course on Mondays and Wednesdays. Will I be recording the lab section? Yes, it will be recorded in the sense that it will be a continuation of this video, but it won't necessarily have a tremendous amount of new content or things, but it will be there either as extra time on the current lecture video that you're currently seeing right now, or potentially a separate video. So I have to work that out just yet. Okay, gang, do you have any questions right now about this unusual time of life? Certainly, when we get to the lab time, which is going to be a combination of lab and office hour time, if anybody would like to talk about what's happening, if you have any questions about the underlying biology of coronaviruses, I'd be happy to talk with you about these things because they're very relevant today. Okay, I have a question. Suzette is asking, how will we be taking our tests? I'm still working that out, but our tests will be entirely online. I'm not 100% sure exactly how I'm going to do that yet, but expect me to do some experiments with your group in the next week or two with a website called classmarker.com. Classmarker.com is an online testing service, and I anticipate putting exams on that website. So the quick answer is all exams, lecture and lab, will be online, and very likely they will be on a website that I will share with you soon called classmarker.com. Okay, any other questions right now? Okay, I'm not seeing any questions right now, so I guess I will begin. You know, I hope you did have a nice spring break. It's kind of like that's a lifetime ago away because of all the chaos and stress that's going on around us. So I want to encourage you to please continue to take care of yourselves. You know, I will not be taking attendance for any of our online sessions. I understand that there's a new reality right now. So if you can't make class, lecture, or lab, I'm not going to give you a hard time about it. But I want to encourage you to try to keep up, particularly if you don't make a class, to at least go see the video so that you get important notes as we go forward. Okay. So if there's no further questions, what I'm going to do, gang, is I'm going to start by summarizing a couple points from chapter 5 about skin. And then in about 20 or 30 minutes, we will wrap that up and we will start chapter 6. There are a few things that I'm not going to cover with you that I normally would cover with you because, you know, we have very limited time at this point, and I need to be sure that I hit the highlights of the chapters that we have to talk about. Okay, so what I'm going to do at this time is I'm going to start sharing my screen with you, and I'm going to bring up a Microsoft Word document. And what I'm going to do is start typing notes into that Word document. I want to encourage you to take notes just like you would take notes if we were in class together. In other words, it's important that you actually handwrite material to learn. Learning is an active process, and so as you write the notes that I'm going to put on the screen for you, it will help keep it in your head. So I want to encourage you to take notes actively during our time together. And also, you know, if I'm going too fast, to please slow me down, speak up, or put something in the chat window so that I know I'm going too fast. The reality of it is I can type faster than you can probably write. So I need to be careful not to type too fast and go like a lawnmower, so to speak. Okay? All right, so I hope you're well, and let's see what we can do with this. Question, will you share the document before the test? I don't have any, well, okay. Let me ask you, would you like me to email you the Word documents after each lecture? Please type yes or no into the chat window. Okay. All right. I will email the Word documents to you after each lecture. Even so, I want to still encourage you to take your own personal notes because, again, I can't emphasize enough that we are not robots in the sense that you can't just watch or listen to something to learn. You actually have to engage your muscles and be active about it. Please take notes during our lecture time together, and then you can supplement your notes with the Word material that I will send to you at the end of each class. Okay. Another question, how is the lab test going to be on the 6th? Kimberly, I can talk more about that in our lab time together, but again, all exams, lecture and lab will be online. Kimberly, I think you took Biology 111 with me, so you should be familiar with the ClassMarker website. At this time, I expect I will be relying on ClassMarker to give you your exams. Okay, gang, let's get started here with content. And again, if you need me to slow down, please interrupt me in some way, and I'll be sure to do that. Okay. So here we go. I'm going to share my screen with you. And you should be seeing a Microsoft Word document. For those that I can see video on, please give me a thumbs up if you can see the Word document. Okay, thank you very much. I'm seeing a thumbs up. Okay. Okay, so I'm putting in Biology 121 and today's date. Okay, gang, what I'm going to start talking with you about is actually content that ends up chapter 5 on the functions of skin. So I'm looking in your textbook at about pages 158, 159. Okay. Okay, functions of the skin. Okay, many of these things you already know about, but again, what I want to do is simply summarize some of the major themes we were trying to talk about before we went on break. Okay. First off, Your skin is heavily involved in regulating your body temperature. The fancy vocab term for that would be thermoregulation. Okay, thermoregulation refers to the homeostatic regulation of your body temperature. Okay, now the thermoregulation has two aspects to it. First, you have glands that are called eccrine sweat glands. These are the glands that actually produce the sweat on your body. So you think about the sweat that may show up under your armpits when you're outside on a warm summer day and you're walking or exercising in some way. Those sweat glands are producing sweat, and as that sweat, which is made of water and some dissolved solutes, as it evaporates off the surface of your skin, you release heat energy that was trapped inside your body and that actively cools your body. Okay, so the eccrine sweat glands produce sweat, which evaporates off the surface. It's that evaporation which actually releases the heat from your body. Okay. The second point of this is that for thermoregulation, your blood vessels have the ability to constrict or to dilate. For example, if you're overheating, if you're retaining too much body heat, then the blood vessels will dilate, which means they widen, they open up. And that allows an increase in the volume of blood flow to the surface of your skin at the dermis, which means that you can release more heat from the surface of your skin once the blood moves to the dermis. Okay. Okay, if you're overheating, blood vessels will dilate, and this allows an increase in blood flow. To support sweat production. Okay. If you're cold, your blood vessels are going to constrict, and you're going to reduce blood flow to the dermis and reduce the amount of sweat that your body produces, reducing the amount of sweat that you can evaporate to release heat. Okay. All right. So once again, if you're cold, blood vessels constrict to reduce the blood flow to the dermis. Okay. So are you guys okay right now? Give me a thumbs up or a thumbs down if you're okay. You're okay. All right. I'm seeing an okay. All right, gang. So the first thing was thermoregulation. The second aspect, second function of the skin is simply the skin acts as a blood reservoir. Okay, your book tells you that the dermis has an extensive network of blood vessels that typically carries about 8 to 10% of your total blood volume during rest. I mean, most of us are honestly just resting right now. You're sitting in a chair, watching a lecture, listening to a lecture. That means about 8 to 10% of your blood volume is sitting in your dermis at this time. Okay, the dermis contains an extensive network of blood vessels that carry about 8 to 10% of total blood volume, while you are at rest. Third function, protection. Okay, there's several aspects to this. All right, first off, remember we talked before break, we talked about the fact that your skin has all these keratinocytes. Well, the keratinocytes contain a lot of keratin, and we recall that keratin is a protein. This protein is very tough and durable. It's almost like the equivalent of chain mesh armor that the knights of King Arthur would wear. It's a very tough protein, and it provides protection to your body. Keratin, a protein, provides physical protection from abrasion. Okay, so it helps prevent scrapes and cuts to your skin. Okay, it provides protection from abrasion. It provides protection from microbes that might want to enter your body. It forms a physical barrier to prevent a lot of bacteria from entering the body. It does provide a physical barrier, at least in a limited sense, to some chemicals that might want to leach into the body. And it also provides some limited protection from heat. In other words, you can touch a warm surface, and as long as it's not too warm, it doesn't damage the internal tissue of the body that's deep to the dermis. Okay. Secondly, functions of the skin. Okay, let's talk about your skin oil. Okay, the skin oil, okay, you realize that particularly during the wintertime months or during our emergency right now, people are being advised to wash their hands a lot. That's one of the best defenses we have against the coronavirus right now. But you appreciate that if you overwash your hands, you can remove the oils from the surface of your skin, and your skin becomes overly dry and cracked. So what I'm saying is that one of the functions of the skin is to provide a lubricating oil, and it's important that you appreciate that that oil actually has antimicrobial properties. The pH of your skin oil is mildly acidic, which tends to be antibacterial in nature. In addition to the skin oil feature of protection, there's some other things we should say. This goes back to our time before break where I was pointing out the cells that you find in your skin. And we'll simply notice that, once again, the epidermal Langerhans cells, which are a type of macrophage that can protect your body from bacterial and microbial invasion. Okay. Epidermal Langerhans cells have phagocytic ability to protect and attack microbial invaders, to protect against and attack microbial invaders. Okay. There's lots of things we could say under the function of protection. I think I'll talk about one more, and that is the fact that we have melanocytes. Melanocytes we talked about before break. Melanocytes are the cells that produce melanin pigment, and it's the melanin pigment in your skin that's capable of protecting the DNA inside your skin cells from ultraviolet radiation from sunshine primarily, and other artificial sources of ultraviolet radiation that could potentially cause you harm. Okay, melanin pigments produced by melanocytes protect skin cell DNA from ultraviolet radiation damage. Okay. All right. Okay, gang. So let's see for a moment. Okay. All right. All right. Let's see. Okay. So, gang, do you have any questions at this time? Okay, I'm not seeing anything, so I'm going to continue on. All right. Please interrupt me by unmuting your microphone if you do have a question. As I look at things on my shared window on the computer right now, I can't really see the chat window, which I would like to be able to see it, but I can't. So if you do have a question, please unmute your microphone and speak a question. So I'm going to ask the group of you a question right now and see if one of you can maybe answer that question. Here's my question for you. I'll type it out as I also ask it. How is DNA damaged by ultraviolet radiation? Are any of you able to answer that? Feel free to unmute your audio if you'd like to try to answer that. Do we have anybody that can do that? Okay. Nobody wants to be brave?
Does it interfere with the ability to reproduce in some way?
Thank you, Brendan. Ultimately, it would interfere with the ability to reproduce. That's true. That's true. And I'll put that down in just a moment. Anybody else? Okay, so I'm kind of seeing my participant list here. All right. Okay, go ahead.
Does it have to do anything with the sugar phosphate backbones?
You're getting close to it. It actually has to do with the nitrogenous bases. If you remember your lessons from chapter 2 about the chemistry of DNA, you remember the chemistry of DNA is made of nucleotides. Nucleotides have three things. They have a phosphate group, a sugar, and a nitrogenous base. Those nitrogenous bases come in four different versions, 4 different flavors: adenine, thymine, cytosine, and guanine. DNA is damaged by ultraviolet radiation because UV radiation causes the thymine nitrogen bases to link together in unnatural ways to form structures called thymine dimers. And as Brendan, you indicated with your response, the thymine dimers can definitely interfere with proper cell reproduction. So let me write that down for you. UV radiation induces thymine dimer formation in the structure of DNA, which interferes with proper cell reproduction. Okay. If you're curious about thymine dimers, you know, during your study time, just Google it. Put into the Google search window thymine dimers, And you'll find, I'm sure, many more things than you ever want to know about. Okay. All right, gang, I'm going to keep on rolling here. We're talking about functions of the skin. Okay, number 4. Number 4 is cutaneous sensations.
Dr. Allen, can you switch your screen again?
Can I switch my screen again?
Yeah, because the window isn't, the Microsoft isn't popping up.
Thank you for saying something. Okay, let's see here. Can you see it now?
No.
I'm able to.
Okay, Brendan, you're able to see it. Please give me a thumbs up if you're able to see it. Okay, so I'm seeing several people with a thumbs up. Okay, so do you have a technical problem, Zuzette?
No, I can see you moving around on your screen. It's just that for the switch to screen share isn't popping up. Maybe. Let me see. Oh, there we go. I found it.
Are you okay?
Yep, I good.
Okay. All right, thanks. All right, gang. Number 4, cutaneous sensations. Okay. You know, because we covered it last time, we went over the structure of skin fairly in depth on page 145. And while you don't have to go back to that, I mean, if you have your textbook, you can quickly do that. But one of the things I pointed out is that particularly in the dermal layer, there is a variety of nerve endings, okay? So you have nerve ending receptors embedded in the skin. These include the tactile discs of the epidermis. They include the corpuscles of touch in the dermis. Let's see, I just misspelled corpuscle there. That should be P-U-S-C-L-E-S. Corpuscles of touch in the dermis. And the hair root plexuses around each hair follicle. I mean, you can appreciate if you touch some of the hair follicles on your arm or something, you can feel that. And that's simply what we're pointing out, is that you have all these nerve endings embedded in the epidermis and dermal layers of your skin. Okay, so these nerve endings collectively work together to give you a variety of sensations. The sensations include touch, pressure, vibration, tickling, warmth, and coolness. You can tell when you're touching something hot versus you can tell when you touch something cold. You might think that, you know, you wouldn't need both kind of temperature receptors, warmth and cool, but the reality of it is, just because you're not touching something warm doesn't mean you're touching something cool. So Mother Nature actually created independent warmth and coolness touch receptors. Okay, number 5 for functions of the skin, excretion and absorption. Excretion and absorption. Okay, so for excretion, okay, when you sweat, you remove primarily water from your body, but you will also remove a tiny amount of dissolved solutes that are in that water as it evaporates from the surface of your body. So sweat removes water, heat, and a small amount of salts, carbon dioxide. That's weird, isn't it? I mean, we thought that you simply breathe out carbon dioxide. That's not entirely true. You actually excrete a little bit of carbon dioxide in your sweat as you as you create sweat at the surface of your skin. So you excrete a small amount of salts, carbon dioxide, ammonia, and urea. Okay, urea is actually a natural breakdown product of protein degradation in the body. It's normal, it's healthy, it's supposed to happen. Okay. Absorption. Okay, for absorption, okay, you might imagine that since your skin cells are cells, right, and they're made of a plasma membrane, which is made of a phospholipid bilayer, that your skin is able to absorb materials that are in fact lipid soluble. Lipid soluble substances have the ability to penetrate the plasma membrane of your skin cells and be absorbed into the body. So certain lipid soluble materials have the ability to penetrate your skin. Okay, this would include vitamins A, D, E, and K. These are lipid soluble vitamins. As well as oxygen. And believe it or not, you can, while you release carbon dioxide from the surface of your body, sometimes a little bit of carbon dioxide is actually absorbed into your skin. It can go back in and out. Oxygen and carbon dioxide, to a small amount, can actually be absorbed through the skin. Okay. This would also include lipid-soluble drugs. Now, there's a whole bunch of those we could talk about. But I'll mention one that's kind of in a joking way, but it's also in a sincere way, right? And that would be testosterone. Okay. Sometimes males, for a variety of reasons, are on testosterone therapy to treat a number of conditions that men suffer from, particularly as they get older. And so one of the warnings typically on these testosterone patches is that you ladies, please do not touch, right? Please don't pick up these testosterone patches with bare skin. Please don't put them on your body, because that drug, testosterone, which is a hormone, which is a lipid hormone, will readily absorb through the surface of the skin and influence your own body's functioning. Okay. All right, gang.
Is it the same for estrogen?
Yes, it is. Okay, estrogen and progesterone. So estrogen, progesterone, testosterone, the quick three hormones that we would think of as sex hormones, are all lipid soluble hormones. So all three of those would readily absorb into the skin. Okay. Do you have any other questions? Feel free to ask. Okay, gang. All right, last thing on this topic, and then we're going to move forward. The last thing on this topic is, okay, so we had number 5, excretion and absorption. Number 6 is the synthesis of vitamin D. Okay. Folks, I'd ask if you please mute your microphones while you're not actively talking, so we don't have background noise for everybody. So synthesis of vitamin D, that's one of the primary functions of your skin. I'm not going to give you a lot of details at this time because it is a major topic of Chapter 6. So I will be talking to you about the synthesis of vitamin D in Chapter 6. But of course, we know that vitamin D synthesis, I'll mention a few things here just to complete your notes. Vitamin D is essential for the absorption of calcium from your food and drink. And of course, calcium itself is very important for how the body functions. And it may be of interest to you know that vitamin D is also a major regulator of your immune system. Okay. Now, we used to teach the immune system in AMP1, folks. That has now been reorganized. You will get the immune system in AMP2. So I won't be talking to you about that this semester. But suffice it to say, if you've had any other biology, you know about the immune system. And you know there's really two branches of the immune system. You have what's called the innate immune system, which your skin is a part of, as well as the adaptive immune system, which is what you might think of when you think about antibodies and B cells and T cells, if you're aware of these things. Vitamin D is a regulator of both branches of the immune system, both the innate and adaptive branches. Okay. All right, folks. I know this is still, it's still not as nice. I can't really see your facial expressions. I can't get a feel of whether you're understanding or not understanding. So you need to let me know if something's bothering you. So before we start the next chapter, let me ask you now, do you have questions on today's notes or anything else from chapter 5? Obviously, I have not literally lectured on every topic in chapter 5, but that's normal for this course. You know that there's not time to lecture on every topic in every chapter. So do you have any questions right now before we start the next chapter?
You said that our skin oil is acidic, but why is it not harmful for us when we keep touching everything that we eat? Like, I know that the oils get onto our food, but why isn't it not harmful to us?
Right. Okay. So your skin oil is mildly acidic. And by mildly acidic, I mean its pH is around 6.5, somewhere in that vicinity. So it's not corrosively acidic like hydrochloric acid. It's not like you see on the horror movies where somebody throws acid on a person and they start to melt away like the Wicked Witch of the West. It's not that acidic, but it is acidic enough for individual microbes that are very tiny like bacteria to have their cellular physiology disrupted from it. It doesn't hurt us very much because as we deposit small amounts of this material on foods, some of that food will neutralize some of those acids before we ever eat those foods. So it's really not a concern for us in that way.
Okay.
Other questions? Okay, gang. All right. So we're going to put chapter 5 to rest and we're going to start chapter 6. Okay, chapter 6 is the bone chapter. Now you folks are all experts on the bones of the skeleton. I mean, you've learned a lot of them. I know maybe some of you had some frustrating results on your first lab exam, but that still doesn't mean you don't know anything. You still know quite a lot, even if maybe your test score wasn't as strong as you wanted it to be. So in chapter 6, we're going to talk about some features of bones, some anatomy features, and we're also going to talk about some physiological aspects of things. Okay, so let's begin chapter 6. Bone tissue. Okay, that is the topic of chapter 6, bone tissue. Okay, so a quick overview. A quick overview of bone tissue are some things that you probably know about, but if you don't, we'll put them into your notes at this time. Okay, first off, your bones provide structure and support. That should not be a surprise. Okay. Let's face it, if we did not have the bones of our skeleton, if we did not have our skeleton, we would be made of a bunch of soft tissue that really wouldn't have any definable shape. We would kind of look like Jabba the Hutt, this big pile of protoplasmic goo that kind of just lied on the floor. But that's not what we look like because we do have a skeleton. Number two. Okay, number two. The bones manufacture the blood cells. Okay, you know this, that your red bone marrow makes both the red and white blood cells. Red bone marrow makes red and white blood cells. What is the process of making blood cells called? Can anybody tell me that? What is the process of making blood cells called? What do you say, folks? Starts with an H.
Homeopiesis?
Okay, you're looking at the right thing. It's called hematopoiesis or hemopoiesis. There's two different ways, two different versions of the phrase. Hematopoiesis, or alternatively, hemopoiesis. Okay, that is the process of making the blood cells. And of course, those blood cells are derived from a specialized layer of stem cells that are found in the bone. Okay. The bones contain stem cells that allow for the production of red and white blood cells. Okay. Number three. You should be aware of that your bones store a variety of minerals in them. So they store minerals. Okay. And here I'm talking specifically about calcium and phosphorus minerals, which are stored in the extracellular matrix of bone. Okay. Not only does your bone store minerals, it also stores triglyceride, which again, you should think of triglyceride as biochemical energy. And so your bones act as a triglyceride repository, or if you prefer, triglyceride battery to store that material. Okay.
Dr. Allen, triglyceride is a lipid, correct?
That is correct, yep. It's got a glycerol molecule and 3 fatty acids. Okay, triglycerides are energy batteries. Okay, all right, this is stored in the yellow bone marrow. Okay. Now, when you talk about the skeletal system, that phrase skeletal system has a specific meaning. When you say skeletal system, you're referring to the entire collection of bones, pieces of cartilage, pieces of ligaments, and pieces of tendons that comprise the system. And finally, I would want you to be aware of the term osteology, right? Osteology is the study of bone structure and the treatment of bone disorders. Osteology is the study of bone structure and bone disorders. Okay. All right, gang. So, okay, I'm looking at my time here. Time goes way too fast as usual. Okay, let me point out a few features of the long bone that you see on page 173. Now, much of this, or at least some of this, you would have had some exposure to in your lab homework when we were covering the skeletal system. So I don't anticipate that all of this is brand new for you. Some of these terms should definitely be ringing a bell in your mind because you would have been exposed to it during your lab homework. So we'll go fairly quickly over this, but there are a few things I do want to be sure are in your notes. Okay. Structure of a typical long bone. So I'm specifically looking at figure 6.1 on page 173 of the current edition of your textbook. Okay, so first off, you appreciate when we say a long bone, that the phrase long bone refers to the fact that the bone has a greater length than width. That's what long bone means. Long bone means the bone has greater length than width. No big deal there. Secondly, let's point out a little bit of the anatomy of the long bone. First, we have the diaphysis. Okay, the diaphysis, if I can spell properly, is the shaft of the bone. And you can look at these things. If you have your textbook available, I would encourage you to have page 173 open. You can look at these things as we put them into your notes. The diaphysis is the shaft of the bone. Number two, you have the epiphysis, or plural epiphyses. The epiphyses refers to the ends of the bone, so both the proximal and distal ends of the bone. Epiphyses, so I'll give you the plural version, refers to the proximal and distal ends of the bone. Okay. We have the metaphyses. Okay, the metaphyses refer to the regions between the diaphysis and each epiphysis. Between the diaphysis and each epiphysis. Okay. These are the regions between the diaphysis and each epiphysis. Okay. All right, and please note, this is where you get, this is where we get the phrase, well, there's a couple things I want to talk about here now that we have epiphyses and metaphyses listed down. Okay, so forgive me for a moment. I'm going to come up between 2 and 3 in the notes here and highlight a couple things, okay? So it's in the epiphysis, kind of, on page 173, where you see that phrase epiphyseal line. Okay, so let's try to disambiguate this concept. In other words, I want to make this as clear as I possibly can. Okay, so if you've ever heard of the phrase cartilaginous growth plate, that is the same thing as the epiphyseal growth plate. Okay, the cartilaginous growth plate is the same thing as the epiphyseal growth plate. This is a section of cartilage from which your long bones grow. So, you know, when you were 11 years old, 12 years old, and you started to enter puberty and you started to get taller, you started to go from 3'10" to 4'7" or whatever it might be. As you started to grow, your long bones started to grow at the epiphyseal growth plates. So this is an area made of cartilage in young, meaning youthful bones that allow you to grow up. Okay. Oh my goodness. Did you just hear my Three Stooges clock? Oh, sorry about that. You're probably going to experience that. I have a Three Stooges clock in my home office. Okay. So the cartilaginous growth plate, which is the same thing as the epiphyseal growth plate, is the area of the long bone that's made of cartilage in young people, children, youth, teenagers, that allow you to grow up. Okay? When you become an adult, that epiphyseal growth plate becomes the epiphyseal line. So you can see on page 173, the epiphyseal line is indicated. If you see the phrase epiphyseal line, that means you're looking at an adult bone that has finished growing. This is the area that was the growth plate, but is now finished growing in an adult bone. This is the area that was the growth plate, but is now finished growing in an adult bone.
Go
Go ahead.
Allen, 2 number 2s.
Oh, thank you. Okay, so I'll change that in the document for you. Thank you. Okay, so the epiphyseal line is not made of cartilage. It's actually made of bone tissue. So what I'm trying to tell you is that as you grow up and change from a teenager to a mature, a physiologically mature adult, the cartilage that's found in the growth plate is actually differentiates into bone tissue, osseous tissue. Okay. Okay. The epiphyseal line is made of osseous tissue, not cartilage. Okay. So I apologize if I'm repeating this a lot, but I really want you to understand this. The epiphyseal growth plate is made of cartilage. The epiphyseal line is not made of cartilage, it's made of osseous tissue. So as you grow up and become an adult, the cartilage actually converts into bone. One last thing, which is kind of a weird thing, if you notice very carefully, if you look very carefully at the picture on page 173, a large part of the epiphyseal line is not in the epiphysis at all, is it? Where is most of the epiphyseal line? When you look at that picture on page 173, look at the epiphyseal line. Is it really in the epiphysis?
No, it's towards the head.
Okay. Brendan, stick with me on that. What bracket along the left would you say encompasses most of the epiphyseal line?
The metaphysis?
You're right, the metaphysis. So it's kind of a misnomer. It's a misnomer that the epiphyseal line is really in the epiphysis. Most of it is actually in the metaphysis. Okay. I will not have a gotcha question on a future exam where you tell me that the epiphyseal line is really in the metaphysis. I'm not going to do that to you. But I want you to look at that carefully enough that you appreciate that while we do call it the epiphyseal line, it was kind of historically mislabeled. The epiphyseal line really should be really called the metaphyseal line. Just one of those kind of gotchas that have occurred throughout history with human knowledge. And you just have to learn to accommodate these things and memorize these gotchas so that you're aware of them. Okay, gang. I'm going to keep going here. Okay. Structure number 5, I want to highlight the articular cartilage. The articular cartilage. Okay, the articular cartilage, which you see on page 173, is a thin layer of hyaline cartilage that covers the part of the epiphysis, covers the part of the epiphyses where the bone forms a joint with another bone. Okay. Okay, a thin layer of hyaline cartilage that covers a part of the epiphysis where the bone forms an articulation. That's the fancy word for joint. With another bone. Okay. All right, so let's continue here. Number 6. Also pay attention to the periosteum. The fact of the matter is that all the bones you learned in lab, you know, when you were looking at the bones on your visible body software, or where you were looking at some of the bones that I would pass around throughout the room, or the skeleton at the front of the lab room, if that was a real bone, each of those bones would have been wrapped in a very special membrane called the periosteum. The periosteum you can see on the right side of the long bone on page 173 there. So please put an eyeball on that. Okay, so the periosteum is a membrane that wraps around the long bone. Okay, and what's significant about it is that it provides an attachment point for ligaments and tendons. Provides an attachment point for ligaments and tendons. And let's see here. It also provides an anchoring surface for blood vessels and nerve endings to invade the tissue. And in that use, I'm not saying the word invade in a negative way. I'm just saying as a way to put the blood vessel into the interior of the bone tissue. Folks, please turn to page 176 in your textbook. Take a look at page 176, figure 6.3. On figure 6.3, along the right side of of picture A, in boldface, you should see periosteum. Okay, and what you notice is that there are arteries and veins colored red and blue there that actually connect to the periosteum, anchor in the fabric of the periosteum, and then penetrate deep into the interior structure of the bone. All right, so B, not only does the periosteum provide an attachment point for ligaments and tendons, it provides an anchoring surface for blood vessels to go into the interior of the bone tissue itself. Okay. All right. Somebody looking carefully at page 176, somebody tell me what kind of material is physically stitching the periosteum to the surface of the compact bone?
Is it the veins?
Not the veins, but thanks for trying.
Is it perforating fibers?
Yes, perforating fibers. I want you to be aware of that, folks. The perforating fibers, which are also called Chpt fibers, are actually bundles of collagen, specially made collagen that connect the periosteum to the underlying compact bone. Perforating fibers, also called Chpt fibers. Okay. Very good. All right, let's wrap up with two quick things here, then we have a new topic to cover. Number 7. Please be aware of the medullary or bone marrow cavity. For that, you can look at figure 6.4 on page 177, or you can go back to page 173, whatever you like. Okay. So just making you aware of the medullary or marrow cavity. Okay. This is the hollow space within the diaphysis that contains the yellow bone marrow containing triglyceride material, and you're also going to find some blood vessels in there as well. Okay, so it's the hollow space within the diaphysis that contains blood vessels and yellow marrow. Finally, one more feature I'd like to point out is the endosteum. Okay, the endosteum is a thin membrane that actually lines the bone marrow cavity. It contains a single layer of bone-forming cells. What I'm saying to you is, it's the endosteum that contains the stem cells that make the cells of the bone itself. A thin membrane that lines the medullary cavity. Okay. Okay, this endosteum contains a thin single layer of cells which include stem cells capable of making the bone cells. Okay. Gang, do you have any questions at this time? Okay, you're quiet, so I'm going to assume you're okay. Remember, if you do have questions, you can bring them up again during our office hour time coming up at 9:30. That's quite okay. All right, folks, I'm going to move next to the histology of bone tissue section, beginning on page 174 of your textbook. Bone tissue histology. Okay. So obviously, excuse me, I got a telephone ring. Hold on a sec. Sorry about that. Okay. Since bone is a tissue, it's made of cells and an extracellular matrix, right? Bone is a compact tissue, and you've learned from chapter 4 that compact is a connective tissue, and connective tissues always have cells and extracellular matrix. As a connective tissue, bone tissue is made of cells and extracellular matrix. Okay. Now let's see. Let's just make a few statements about the extracellular matrix, and then I'll focus on primarily the cells we find in the tissue. The extracellular matrix of the bone has mineralized salts in it, primarily calcium and phosphorus, and these mineralized salts are what provide the hardness to the bone. So the bone hardness is due to the presence of crystallized mineral salts like calcium and phosphorus. Okay, so we'll underline the phrase hardness. The bone hardness is due to the calcium and phosphorus salts. And the bone's flexibility, the fact that they can bend a little bit before they break, is actually due to the presence of collagen fibers found throughout the matrix. Bone flexibility is due to the presence of collagen fibers. Okay, in many ways, the analogy that some of you have heard me used on this is that the extracellular matrix is similar to steel reinforced concrete, where the steel provides flexibility to the concrete. So the collagen fibers would be the steel in my analogy, and the hardness of the concrete is due to the concrete itself, which in my analogy are the mineralized salts, the calcium and phosphorus. So for the extracellular matrix, think steel reinforced concrete. Okay. All right. Please let me know if you have questions about that, gang. What I'm going to do is focus on some of the cell types you see on figure 6.2 at this time. Okay. So cells in the bone. All right. First, we have the osteoprogenitor cells. Osteoprogenitor cells. These are also called osteogenic cells. Okay, these are also called osteogenic cells. And what I'm trying to tell you with these is that it is the osteoprogenitor cells which are the bone stem cells. These are the stem cells of your bones. Every other kind of cell that you will find in the bone, every other kind of bone cell that you find in the bone originates, has an ancestry that relates them to the osteoprogenitor cells. Just like you are related to dad, and you're related to grandma and grandpa and great grandma and grandpa, all the bone cells you find in the bone have an ancestry or cellular lineage that connects to the stem cells, which are properly called the osteoprogenitor cells. These are the cells that are found in the endosteum that I highlighted in your notes just 5 minutes ago. Okay, so the osteoprogenitor cells are the stem cells of your bones. Okay, next we have the well, before I leave the osteoprogenitor cells, let me make one other thing perfectly clear. Okay, it's the osteoprogenitor cells of the bones. Since they are the stem cells, you do appreciate that these are the only kind of bone cell in your bones capable of reproduction. In other words, they're the only kind of cell in your bone that are capable of mitosis. All right. Gang, I am shocked that it is now 9:15. That means we have to pause because that's all we're scheduled for, and I appreciate that some of you might have other obligations. Okay, so at this time, what I'm going to do is take a 15-minute break. I'm going to leave this video Zoom session live, and I will be back online with you at 9:30. If you want to stay for that, you may. Again, you're not obligated, but it's during the 9:30 time that I can give you answers to your lab homework and also potentially provide support for other things you may need help with. So, do you have any burning questions you want to ask before we take a 15-minute break?
I have a question.
Go ahead.
For the homework, will that still be quote unquote due every Monday if we're not in lab?
Technically, no, since I have no way of collecting it from you to look at it. I mean, I thought about this long and hard. I thought about, well, I could have you take pictures of it and send me all the digital pictures, but that was just going to be a logistical nightmare. So I decided not to do that. So you don't have to hand things in to me until we return to school, but I'm going to encourage you to stay on the schedule that's listed in the syllabus because it will help you pace yourself so that you don't get overloaded. You don't want to get in the habit of not doing these things, and then 4 weeks from now we go back to school and then I ask for like 6 assignments and you don't have them. That would be a nightmare for you. So no, you don't have to stay on the schedule because I'm not technically collecting them at this time, but I'm going to encourage you to try to stay on that schedule for your own personal sanity.
Okay.
Any other questions? Okay, gang, let's take a 15-minute break. I'll be back online with you about 9:30 or 9:35. Okay, go get a refreshment, use the bathroom. Okay, everyone, welcome back. As I mentioned earlier, this sort of second hour is meant to be sort of a support hour for both lab and lecture content. In some ways, it's a little fortunate that, I have you learning a lot, really most of your anatomy on the visible body software. So if you haven't made that investment yet, I think it's almost critical that you do that to get through the rest of the semester, because we just don't know when we're going to get back to school, or if we'll be back to school at all, to be honest with you. I think it's somewhat unlikely that we will get back to school this semester. So you have your vocab list for the muscles that are due, that you're responsible for. And what I can do at this time is on a weekly basis go over some of the lab homework answers, as we always do with the reviewing your knowledge and using your knowledge sections, and then open the rest of the time for any conversation or questions you might have about either lecture content or lab content. So what I'll do at this time, please give me a thumbs up if you can hear me. Okay, thank you. Let's take a look at your lab homework that was due today. Obviously, I would normally have you exchange it with a neighbor and you would circle each other's incorrect answers. Do the same thing for yourself today. Just circle things that are wrong that you need to go back to and review. Let's take a look at lab exercise 11 and go to the reviewing your knowledge page 171. Page 171, reviewing your knowledge, exercise 11. And what I will do is read down the answers for you so that you know what should have been correct there. Okay, we're going to go over part A, structural and functional classification of joints. Number 1 was a diarthrosis. 2, synarthrosis. 3, amphi, A-M-P-H, amphiarthrosis. 4, synovial. 5, cartilaginous. 6, synovial. 7, fibrous. Moving to letter B, basic structure of synovial joints. Number 1, hyaline. 2, synovial. 3, you could have said hyaline or articular cartilage. 4, bursa. 5, synovial membrane. Letter C, types of synovial joints on page 172. Letter C. Number 1 is a pivot joint. 2 is a ball and socket. 3 is a hinge joint. 4 is a planar, which is also sometimes called a gliding joint. 5 is saddle. 6 is condyloid. Letter D, movement at the synovial membranes. Number 1 is adduction. That's A-D-D, A-D-D, adduction. 2, extension. 3, depression. 4, supination. 5, plantar flexion. 6 is flexion. 7 is abduction, A-B-D, abduction. 8, rotation. 9, elevation. 10, protraction. 11, inversion. Okay, gang. If you need me to repeat any of those, please type your needs into the chat window. Okay, I'm not seeing any, So I'm going to go to using your knowledge, which is on page 173. Letter A, the synovial joints. Okay, letter A, number 1 is articular cartilage. 2 is the synovial cavity, which also can be called the glenoid cavity. 3 is the articular capsule, which is sometimes referred to as the fibrous capsule. 4 is articular cartilage, or you may have referred to the trochlear notch, and that's okay. Number 5 is articular cartilage, or the fibrous capsule. 6 is the acetabulum. 7 is the synovial cavity. 8 is the greater trochanter of the femur. 9 is ilium, and 10 is the head of the femur. Moving to section B on page 174, the synovial joint movements. Number 11 is flex. Number 12 is dorsiflex. Number 13, extend. 14, extend. 15, plantar flex. Okay, I'm seeing some requests for repeated things on the chat window. Let me take them one at a time. Okay, Brendan, you said 5 through 7. What part are you talking about? Are you parking letter D there?
Yeah, I posted a little bit later D5 through 7.
Okay, thank you. Okay, so we're just reviewing some of the things we've already said. Letter D, movement at synovial joints. Number 5 is plantar flexion. 6, flexion. 7, abduction, ABD, abduction. Okay, Allie, for section B, number 4, that was bursa. B4 is bursa. Kimberly, you wanted number 11. I'm going to assume that's section D. So D11 was inversion. Jennifer, you're asking for using your knowledge, section A, 2, 4, and 5. Okay, section A, using your knowledge, number 2 is synovial cavity or glenoid cavity. Either of those is fine. Number 4 is articular cartilage or the trochlear notch. Number 5 is the articular capsule or fibrous capsule. Anything I missed, gang? Anything you would need me to repeat again? Okay. Section B, numbers 1 and 3 for reviewing your knowledge. Okay. Section B, reviewing your knowledge. Number 1 is hyaline. 2 is synovial. 3 is hyaline or articular cartilage. Anything else you need me to repeat for exercise 11? Okay, using your knowledge, 6 and 7. Okay, using your knowledge, number 6 is acetabulum, right? That's ACETABULUM. And 7 is synovial cavity. Using your knowledge, 8, 9, and 10, section A. All right, section A, using your knowledge, 8 is the greater trochanter of the femur, 9 is ilium, and 10 is the head of the femur. Okay, gang, let's take a look at exercise 6. Let's take a look at exercise 6, and let's go to reviewing your knowledge on page 77. Page 77. Okay, I'll give you a chance to get your exercise in front of you. Exercise 6, reviewing your knowledge, page 77. Okay, so for lecture exam 1, you had to know some basic properties of tissue. You remember the essay at the end of the exam. For lab exam 2, I'm going to start tickling you in a bad way about identifying tissues, to look at the appearance of them and know what it is you're seeing. That is a harder skill. Let me say this up front. There's going to be muscle identification and tissue identification on your next lab exam. But if you have to prioritize what you know, you should know the muscles. There will be far more muscle identification questions than there will be tissue identification questions. So I know with life being a little unusual right now, you might have to prioritize where you put your energy. If you have to do that, put your energy into knowing the muscles. Okay, reviewing your knowledge, page 77, letter A, primary tissue structure and function. Number 1 is nervous tissue. 2 is muscle tissue. 3 is connective tissue. 4 is epithelial tissue. 5, 6, and 7 are epithelial, muscle, and nervous. Questions 8, 9, and 10 are epithelial, muscle, and nervous. So the same thing as the past 3 questions. Question 11, connective. Question 12, connective. Question 13, epithelial. Question 14, nervous. Question 15, muscle. Okay. Any of that you want repeated?
What was 11?
Number 11 was connective tissue. Anything else? Okay, gang, let's go to section B on page 78, epithelial tissues. Letter B. Number 1 is stratified squamous epithelium, stratified squamous. Number 2, simple squamous. Number 3, simple cuboidal. Number 4, pseudostratified ciliated columnar. Number 5, simple squamous. Number 6, simple columnar. 7, transitional epithelium. Any parts of section B you want repeated? Number 6.
What was number 4 again?
Okay. Number 4 was pseudostratified ciliated columnar. Zuzette, number 6 is simple columnar. Okay, moving on to section C for the connective tissues. Number 1, elastic tissue. Number 2, dense regular tissue. Number 3, hyaline cartilage. Number 4, areolar. Number 5, elastic cartilage. Number 6, spongy bone, which is also called trabecular bone. So 6 could be spongy or trabecular bone. 7 is reticular. 8 is dense irregular. 9 is blood. 10, adipose. 11 is compact bone, which is also sometimes called cortical bone. So 11 could be compact or cortical bone. 12 is fibrocartilage. 13 through 18 are areolar, reticular, adipose, dense regular, dense irregular, and elastic. 19 and 20 are compact bone and spongy bone. 21, 22, and 23 are hyaline cartilage, elastic cartilage, and fibrocartilage. 24 is blood. Okay, folks, any part of C that you would want me to repeat?
Can you repeat 12, please?
Okay, 12 is fibrocartilage. Anything else? I'm looking for number 4. Osena, number 4 is areolar.
Could 24 also be plasma, or is it just blood?
Hold on one moment here. Let me take a look at it. I would accept plasma. That would be okay. Anything else? Okay, gang. Let's go to section D, the muscle and nervous tissue. Okay, page 79, letter D. Number 1 is smooth muscle. 2, skeletal muscle. 3, cardiac muscle. 4, nervous tissue. 5 and 6 are both smooth muscle.
For number 4, would you accept neurons?
Well, you would certainly get partial credit for that, Kimberly. Probably not whole credit, because a neuron is a type of cell. It's not a type of tissue. And so they're asking you for the tissue, so nervous would be a better answer. Anything else? Okay, gang. Let's do letter E. Here in your lab manual, page 80 and 81, you had a bunch of pretty pictures and you had to see what you could make out of them. Okay. Letter E, page 80, number 1. Number 1 is pseudostratified ciliated columnar epithelium. Once again, pseudostratified ciliated columnar epithelium. Number 2, stratified squamous epithelium. Number 3, transitional epithelium. Number 4, simple columnar epithelium. 5, here comes the Stooges, hold on. All right, sorry about that. I might have to take my Three Stooges clock out of the office. Okay, let me back up. Number 4, simple columnar epithelium. 5, simple cuboidal epithelium. 6, simple squamous epithelium. 7, dense regular connective tissue. 8, compact bone. 9, elastic connective tissue. 10, areolar connective tissue. 11, hyaline cartilage. 12, adipose tissue. 13 is blood. 14, dense irregular connective tissue. 15, reticular connective tissue. 16, elastic cartilage. 17, spongy bone. 18, fibrocartilage. Okay, so I know that was a lot, and there's some big, fancy, complicated terms here. Any of part E you want me to repeat? Number 15. Okay, 15 is reticular connective tissue, reticular. Numbers 9 and 10 are elastic connective tissue for 9, and 10 is areolar connective tissue.
Can you repeat 12, please?
Okay, number 12 is adipose tissue. Any others from section E you'd like help with?
1 and 16.
Okay. Number 1 is pseudostratified ciliated columnar epithelium. Once again, pseudostratified ciliated columnar epithelium. And number 16 was elastic cartilage. Any others from section E? Okay, let's finish off section F on page 82, section F. Number 1. I have a question here. Are we going over the other 2 packets? Which other 2 packets are you referring to, Jennifer? Muscles. 1 second. Let me look at my original syllabus. Yeah, even with the original syllabus, Jennifer, the only 2 lab exercises that were due are exercises 11 and 6. So if you're talking about exercises 12 and 14, those wouldn't technically be due until next week. So we will go over them next week. Okay. All right. Letter F, number 1 was smooth muscle, 2 nervous tissue, 3 skeletal muscle, and 4 was cardiac muscle. If you look carefully at number 4 there, you can clearly see the intercalated discs. They're like, on that picture, they're horizontal, deep pink, purplish lines that you can see in between 2 cells. All right, gang. Anything from reviewing your knowledge that you need repeated? Okay, let's go to using your knowledge on page 83. Okay, Now here, it's very unlikely that your answer will match mine word for word because you had to create your own answer. So don't grade yourself based on whether you have exactly what I say. Look for common ideas and themes. Make sure you have the basic idea down, and that's sufficient. Okay, so for section A, they were talking about genetic diseases and tissue functions. So for each disease, you had to identify the abnormal protein and state how the abnormal protein affects the function of the tissue. Okay, number one, the abnormal protein is a very long phrase here. Here it is. Cystic fibrosis, transmembrane, conductance, regulator, protein. Once again, cystic fibrosis, transmembrane, conductance, regulator, protein. What happens with that disease is that the protein transports chloride ions across the plasma membrane of epithelial cells in the mucous membranes. Okay, that's what it does. That's what the protein does. If you have a defective transporter protein, that lowers the secretion of chloride ions, which increases the reabsorption of sodium and water across the epithelial cells. There's an osmotic principle happening here. The increased reabsorption of sodium and water from the mucus causes the mucus to be abnormally thick. So certainly your answer should have mentioned the presence of abnormally thick mucus. The thicker mucus increases inflammation in the body, and it promotes a risk increase of infection in the lungs, the pancreas, your GI tract, and can sometimes even clog some of your exocrine glands. Okay, so with cystic fibrosis, you have a defective transporter protein that leads to accumulation of mucus, which causes an increased risk of infection and inflammation. That would be the general ideas that you should have in your answer. Questions? Okay. Number 3, we're talking about, okay, so the first 2 questions were about cystic fibrosis. 3 and 4 are about Duchenne muscular dystrophy. Okay, number 3, the abnormal protein is dystrophin, D-Y-S-T-R-P-R-O-P-H-I-N, dystrophin. Okay, when you have a defective dystrophin protein, that causes the plasma membrane of the muscle cell, which is called the sarcolemma, to actually tear. It physically rips. And when that happens, you weaken the muscle contraction that occurs, and that can lead to death of the muscle cell itself. As you have muscle cells dying, you lose muscle mass so that your muscles in your entire body begin to atrophy. Obviously, if your muscle in your heart weakens, or if your respiratory muscles for the diaphragm and the intercostals between the ribs of the rib cage weaken, you get to a point you may not even be able to breathe anymore. So it's a pretty nasty disease. Truth be told, geneticists have discovered and identified over 50 different mutations in that dystrophin protein, meaning there are over 50 different varieties of muscular dystrophy defects. And it's a really difficult disease to combat. Okay, questions? Okay, letter B, nutritional deficiencies in tissue function. Number 5, vitamin C. Number 6, we have to describe the extracellular matrix changes and the symptoms. Okay, if you have a deficiency in vitamin C, your body will experience poor collagen formation, which results in a loosening of your teeth. Your teeth get loose. You will have an increase in bleeding, and you will have problems with any wound on your body healing. You will have poor wound healing because of vitamin C deficiency. Okay. All right. Okay, gang. Page 84, the tissue forming organs. First off, let me say for question C, I think question C is a little bit on the ridiculous side. I would never ask any question like this of you. These were hard questions. They really were. And so I don't expect that you necessarily got everything, and that is okay. Okay, so don't worry about getting a question C type question on a future exam, because I will not do that to you. But just so you've heard the answers once, I will read the answers to you. So we are identifying a tissue in order from the lumen, the hollow surface, to the outer surface. From the lumen to the outer surface. The esophagus, number 7. Here we go. Again, it is totally okay if you didn't get all of this. Number 7. First, we have non-keratinized stratified squamous epithelium, which bumps up against the areolar connective tissue. You may have written down lamina propria, depending on how much you dug into this. Next is the smooth muscle, which is called the muscularis mucosae. Next is the areolar connective tissue, followed by the skeletal muscle in the upper and middle portion, and smooth muscle in the lower portion of the esophagus. Again, it's totally okay if you didn't get these. Number 8, the small intestine. Small intestine, we begin with the simple columnar epithelium, followed by areolar connective tissue, followed by smooth tissue, followed by the duodenal glands, followed by smooth muscle, areolar connective tissue, and simple squamous epithelial tissue. I know that's a lot. You're not going to get that on a test. Don't worry about it. Number 9 is the trachea. For the trachea, you would start with pseudostratified ciliated columnar epithelium. Then you'd go to the lamina propria, which contains elastic and reticular fibers. Then to the areolar connective tissue, then to hyaline cartilage and areolar connective tissue, which for the trachea would have been called the adventitia. And then number 10 is the ureter. Okay, the ureter, going inward to outward, we start with the transitional epithelium, followed by areolar connective tissue, followed by smooth muscle tissue, followed by more areolar connective tissue. Okay, so C was almost an exercise in insanity, and I really don't want you to worry about exercise C. That is not an important consideration for you at this time. Okay. Gang, do you have any parts of A or B that you would want to ask a question about? Okay. So let's recap what's going on at this time. All right. So your next lab exam for the Monday group, and honestly, since we're all testing online at this time, you could take the exam on Monday or Wednesday. It really wouldn't matter to me. It's going to be basically in 2 weeks. So 2 weeks from today, you have to be ready to identify muscle, and you have to be able to identify some tissue. Muscle identification is far more important than tissue identification at this point. You will probably have about 30 to 35 muscle identification questions and probably about 5 tissue identification questions. That's how it's going to break down. Now, normally, if I was with you in class, in lab, I would hand out an additional histology study packet for you as homework. And I am going to do that by way of sending you the histology packet through email. And right now I'm simply trying to locate that in my personal notes. Hold on one moment, please, and I'll see if I can find it. Yeah, okay. All right, there. Okay, so if you have the syllabus in front of you, you know that next week, lab exercises 12 and lab exercise 14 are technically due in the sense that I want you to be keeping pace with things. Lab exercise 12 and 14 go over a lot of muscle anatomy, and I think will be helpful for you to do this. Okay? I'm not grading them right now. I'm not collecting them in any way, but I'd like you to keep up if you are able. It will be in your best interest to do so. I'm going to share my screen with you again and show you the histology handout that I intended to give you in lab today, but which I'm going to have to send through email in some fashion. Actually, it's a very large document. I might need to find a way to give you a Dropbox link to get it because it's pretty big. But look for something from me by the end of today that has a histology handout packet for you. Here's what that is going to look like as I share my screen with you. Right now, you should see a screen called page 1 that has a weird looking pinkish purple tissue with some arrows and brackets in it. Please give me a thumbs up if you see that. Okay, good. All right. So gang, what I'm going to want you to do at home when I send this to you is work through this packet to try to identify everything that has a bracket, a box, or an arrow. Some things will be very easy. Again, you want to look at chapter 4 in your textbook because chapter 4 is your histology chapter. You want to go to chapter 4 in your textbook and use that as a guide to help you with this packet, okay? That being said, some will be very easy and some will not be so easy. So let me give you an example of things. Okay, so for example, page 7 here. Can any of you tell me what this tissue is? So bracket A is around the entire tissue. Do you know what this is?
Bone, compact bone.
Thank you very much. That is compact bone. So the idea is when you get this packet at home, hopefully you can print it out. In a perfect world, you'd have color available, but if you don't, you print it out in black and white, and you go through this maybe looking at the color on your computer, and I want you to identify as much as you can. So for letter A, the bracket, you would write down compact bone. Next to it, I have an area where you can write some notes about the function or the purpose of compact bone. Please note on your lab exam, I will not ask you about function or purpose of the tissue. I will simply ask you, what is it that you're looking at? Identify these things. So letter A is compact bone. Now here's B. B is a box. Can anybody tell me what the box is around?
An osteocyte?
Okay, thank you for trying, Brendan. It's not an osteocyte, but it's an osteon. O-S-T-E-O-N, an osteon. Okay, so that's the idea, gang. For every page, I give you a different tissue, and I want you to identify as many characteristics on each page as you can. Take each letter, that's going to be a bracket, an arrow, a box, and try to identify what it is. And next week, during our office hour time together, I will give you the answers to these things. But you want to do as much as you can, so you learn as much as you can, so you can do well on your lab exam in 2 weeks. Okay? Do you have any questions about the histology packet and what I'm asking you to do? I know it's a lot, and I also appreciate it's a lot at a time in our lives when we're all kind of distracted because of other important things happening. Do the best you can. That's all anybody can ask of you. Okay, so I'm going to stop sharing my screen, go back to the normal Zoom environment. Gang, it's already 10:20. I'll stick around with you for another 20 minutes or so, answering questions, chatting with you, letting me know, let me know where I can help you or what you need support on. Now's the time for you. You can either type something into a chat window, you can unmute your mic and just say something for the benefit of everybody. Whatever you need right now, I'm going to open the floor to you. What do you need right now?
If we have to take our lab exam online for the muscles, I'm a little afraid that like to be able to identify is going to be a little difficult because you're not using the same software that you would be using if we were in class.
Okay, let me calm your fears on that, Kimberly, because I am going to use the same software. I have the software on my home computer, and I have designed the questions from that same software. So it's going to, the pictures and images are going to come from visible body. Of course, they may look different from the viewing perspective that I choose to give you the question, but that's true whether we're in class together or sitting here on a computer, but they will be from the same software.
Will we be using the same website that we would be using the lecture exam to do the lab exam?
That's my intention. There's a few technical details I have to work out in the next couple weeks, but yes, that is my intention.
So would the tissue identification we have to do on the lab exam be something like 80 through 82? I know you said there's only going to be like 5 questions, but would we have to know the difference between like a simple cuboidal epithelium versus like a pseudostratified, or is it just telling the difference between like epithelium and muscle tissue?
Okay, let me clarify for you, Ashley. Because I'm giving you this histology packet that will be coming out today at some point, I'm going to hold you responsible for identifying everything in the packet. So a short answer to your question is you have to know you will have to know anything that's in the packet that I give you.
Will you be giving us a link on like the login and password for then when the test comes to log in?
Yes. There's still a few technical things I need to work out. And you might find that I send the class an email saying, hey, gang, I need your help with something. I need you to test out this technology, this website. And so I would ask you to please help me out with that, because I might have to try a few options to get where I need to be so that I can do this smoothly with you. But yes, the idea would be either as you experienced, as you said in 111, I will either have a class marker thing that opens up naturally at 7 a.m. or I send you some sort of web link through email that you click on to get access to it.
Will you give us like a week before the test? I know the test is in 2 weeks, so like next week, would you give us like an example? Like once you get the website up and running, like to do the test in 2 weeks, will you give us an example test, like 3 questions, just so we know exactly how it's gonna be?
Yes, I will, Kimberly. if I can work that out with the technology I'm reviewing right now, I will. Otherwise, they'll simply manually show those images to you here at an office hour meeting, those same 3 questions, and give you a practice quiz that way. But yes, one way or the other, I will give you a small practice quiz next week. So gang, in some ways, I'm very sorry that, life is kind of dumped on all of us, and even more than usual, you kind of have to be a self-motivated learner, right? You have to study. I mean, you've always had to study, but now I can't be with you to see where the weaknesses are as well. And so I want to encourage you to, if you have questions, to email them to me. Or if you have questions, write them down and ask them during one of our time sessions together here online. So maybe everybody in class can benefit from an answer to the question that you have. I need you to help me know where you need support, because for most of you, I can't see your face. I can't see how you're frustrated about things. So again, this is open time. I'll stay with you for another 15 minutes, and then we'll probably sign off. You may come and go for these meetings as you wish. I'll stay on for another 15 minutes if you have other questions. Content, concerns, coronavirus, whatever you want to talk about, I'll talk about it.
Do you believe that the coronavirus is actually going to get worse and we aren't going to end up back in class?
Well, it is hard to predict things, but personally, I think there's a very good chance that we will not meet again this semester. There is evidence that the coronavirus circumstance is accelerating in the United States, but it's not going to accelerate at the same rate in different areas. For example, things are exploding right now in New York. Things are not so bad in Pennsylvania. So we could have a scenario where they continue to explode in New York, and as the situation in New York begins to stabilize, other areas of the nation, including Pennsylvania, could get worse. There is scientific evidence to suggest that people that have the virus in their body maintain that virus in their body for up to 30 days. Meaning, even if a person is not experiencing symptoms of illness or disease, they could still be carrying that those virus particles in their body and potentially be transmitting them out to the environment for others to pick up for as much as 30 days. Since we know that, it would seem a little bit of a poor decision on the part of our leadership to open school back up in 2 weeks. I think that would be a bad choice for them from a public health perspective. But we'll see what life brings us. But personally, I'm very skeptical that we're going to meet again as a group. And so I'm going to do everything I can to help you get through the rest of the semester with an online teaching modality. It's not optimal, but it's what we have going on right now. Do any of you know anybody that's been diagnosed with coronavirus or that has a potential exposure to coronavirus? Because I do. Yeah. Right? Keep in mind that when you see the news, you tend to see the worst of the worst. Remember, far more people get this virus and experience mild to no symptoms and do not get horribly ill than those that do. Far more people will not get horribly sick from this than do. So keep that in mind when you see all the scary news stories. Most of us will be just fine.
Is it true what they're saying about like how someone with O blood type won't be affected as someone that has like a B blood type?
I haven't heard that, Suzette. I'm a little skeptical of that. I haven't seen any scientific data to support that. That's all I can say, really.
Dr. Allen, about the test, the website you mentioned, I never used that and I don't know how it goes. I was wondering, can you give us more information about that? Because my experience with the online course is like the professor put the test for a certain date, and anytime like during that date we can take it.
Yes, Masena. It would be very similar. Expect to see an email from me in the next couple of days to test some of these new websites and technology out. But the general idea would be exactly what you stated, where you'd be taking a test at an online location, and you would probably have 24 hours to take the test at your home or wherever you're staying at this time. And you know, particularly for lecture exams, since I do have some essays, the computer won't be able to grade those sorts of things. I'm going to have to manually log in and grade them student by student. So it'll still take me a while to get your grades back. But don't be afraid, don't be scared of it. I'm going to give you guys some practice and exposure to it before you have to do the real thing.
So is that going to be like an open book test?
The reality of our current circumstances is that there's no way I can prevent you from using your textbook or notes. I can't prevent it. And so I'm not going to try to. I have the expectation that you may have notes or textbook in front of you when you take your tests, and I'm okay with that.
Is a lot of the lecture exam, is it going to be like multiple choice? And also the same question for the lab exam, is it going to be multiple choice? Or for the lab exam, you're just going to do the same thing you did for the first lab exam? You're going to show the muscle and tell us what tissue it is. You're not going to give us a multiple choice.
Right. It will be like you had in class. You're going to have to actually type in the correct answer to the tissue or muscle. It won't, for the lab exam, it will not be multiple choice. You'll have to tell me specifically what the correct answer is. For lecture exams, you will continue to have a variety of multiple choice, fill in the blank, and if I can work it out, some essay questions that I have to go in and review to grade.
Are you going to also give us for the lab exam a jumbled word bank like you did for the bones?
No, and the reason I won't is because you already have your organized word bank, and I have no way from taking that from you. So we're not going to worry about those kinds of things. You may use the word bank that you're studying from to take your tests, but keep in mind that the lab exam will still be timed. You won't have an unlimited quantity of time. You'll still be given about a minute and 5 seconds or so per question to do. So there still will be that time aspect that you have to prepare for.
How long for the lecture exam would we have? Will we have the same like class time, the hour and 15 minutes to take it?
I think you know, this is not set in stone, and I might change my mind on this, but I'm thinking that I would probably give about an hour and 30 minutes for the lecture exam.
One more question for the final. I know that it says that that we end class the 9th and then the finals start on the 11th. If we had prior arrangements because class was supposed to end basically earlier and we're like a week ahead, can we take the final exam on the 11th if we need to?
I think something like that probably could be worked out. And I would ask you to email me about your concerns, Kimberly. So gang, we don't know what the next 8 weeks hold for any of us personally or our families and loved ones. You know, if you get sick or you have to care for someone that gets sick, please understand I'm not going to give you a hard time about attending class online. Okay, you are adults, you have responsibilities to take care of. I'm not going to give you a hard time about that. But please understand, at a minimum, I do have to hold you responsible for taking exams and meeting those objectives in course. Okay, so you have a lot of latitude right now of how you use your time. You've been through both a lecture and lab exam, so you know how challenging they can be. And I want to encourage you to use your study time as a way to distance yourself from the chaos around you. Right? Everything happening right now is very upsetting. It's upsetting for everybody, and it's hard to focus. But try to set some time aside each day to do some meaningful study in your courses. It can help keep your peace of mind and your sanity by having something that is normal to focus on. So what did you folks do over midterm break? Did you get to do anything fun? Did anybody travel at all?
No.
No traveling?
I worked all week.
The most traveling I did is my son had a birthday party in New Jersey to go to. So we spent a weekend in New Jersey at a birthday party, which was fun. But other than that, it was normal life, taking care of things at the house. So how about the rest of you? Anybody go to a movie?
No, honestly, even now, I'm afraid to go outside because I have kids. So I'm afraid if I get that virus, maybe something not happening to me. But if I bring it to the house, especially I have like a 6 months old baby, so I don't want to make them sick and just stay at home with them.
I can understand that, Mosena. Right, there is a lot of concern right now. I had to go out to the grocery store yesterday, and of course you try to be as careful as you can, but it's very hard to defend yourself against a microscopic virus you cannot see with your eyes. So
I took my oldest to go fishing. That's as far as I went.
fishing's cool. Did you catch anything?
He caught a really big catfish, and I caught a very small bass.
Okay, very good, very good. I see your comment online. You work at a grocery store. Can I come get some toilet paper? All the grocery stores I've been at have no toilet paper. I'm teasing you. I hope you'll be safe.
You can find some at Costco. They have a lot and they limit the purchase so everyone can have.
Okay, that's good to know. Yeah, there's a few places I might try. We're okay right now. I told my little boy he might have to use a leaf outside, but that's okay. So.
I actually called ahead to Target to ask when their truck was delivering. So then I knew exactly when to go and what time to go to get toilet paper before people started going crazy.
Yes. It's at times like this we see the best and worst of humanity. So. Okay.
I just want to know what people really thought about taking all the tissue from everybody. Like they weren't allowed to leave their house basically, and that's all they needed.
Yeah, when people get scared, they act irrationally. They do things they wouldn't normally do, and I think we're seeing some of that. So hopefully that will calm down in the next week or two, and we'll get back to a different new normal for ourselves.
I actually read something that a community broke into a man's house and stole all the toilet paper that he bought out of a grocery store.
Yeah, I'm sure that's happened. I've heard that people stopped by the rest stops along the highway and stole toilet paper from the rest stops. I mean, it's just insane.
Dr. Allen, just a question about the coronavirus. Like in the air around us, there is like food particles. There is like particles we can't see. Does the coronavirus move around in the air like that, or is just like sitting on the surface, and then if we touch those stuff, and then we possibly get it
Yeah.
in our body?
What's making this pandemic so difficult is that it does both. The virus particles will stay in the air within water droplets, and it will also stay on surfaces like stainless steel for about 3 days. So think about doorknobs and places like this. If you've been out, when you come back in, you want to make sure that you wipe down doorknobs that you use to get back into the house. So this particular virus is creating a huge problem for medical officials because it is airborne. It stays in the air when somebody sneezes or coughs, stays in the air for hours, and it will stay intact on certain surfaces for up to a couple days. So you can get it by inhaling mucus droplets that somebody else expelled from their body. You can also get it by touching something that has a mucus droplet on and then touching your eyes, your nose, your mouth in some way. So it's very, very hard to avoid this virus.
You know that there was a movie that came out years ago called Contagion, and it's somewhat what's happening now, that like a pig ate like a bat or something, got infected, and then humans ate that pig, and then this big virus like swept around the world.
Yes, I've seen that. It had, didn't it have Gwyneth Paltrow in it?
I think so.
Yeah, yep. I mean, I saw that movie and it alarmed me because I thought it was pretty realistic. Okay, now we don't want to sit here and scare everybody to death, but the reality of it is certain public health officials have been warning the government for a very long time that we need to take better action to prepare for these kinds of events because they are going to happen. In my microbiology class, I teach a section on the 1918 flu pandemic. Now, what we're dealing with now is not the flu, it's a different virus, but it's behaving in many ways that are similar. And so we've known since 1918 as a society, we should be preparing for things. We should have many, many more ventilators available than we currently do. But as the politicians are, they rarely put resources into things that might only happen once every 50 years or once every 100 years. And now we're paying the price for that.
Could somebody weaponize this kind of virus?
Yes, undoubtedly, yes, if you had the right technical background and lab resources to support it, most definitely, yes. Okay, gang. Well, I think we will sign off. I have a few things I need to do, and I will be sending an email out later to you today with some links to things like the video we have here, and maybe some other information. So look for, typically look for an email from me after each class period sometime that day, and we'll continue from there. Next class, expect me to hit more on bone physiology, particularly how the bones and skin are involved in vitamin D synthesis. And again, please keep your questions, email them, and we'll do the best we can to support you, okay? So take care of yourselves. Don't let worry and anxiety ruin your time off, and do what you need to do to stay safe. And I will see you on Wednesday. Bye for now.